Welcome everyone to this IMAG Multiscale Modeling webinar. My name is Grace Pang and I welcome everyone to this webinar. I'm going to have Herbert Sarro introduce the speaker now on behalf of the Model and Data Sharing Working Group.
Thank you, Grace. Good morning, everyone. Gives me great pleasure to introduce Phil Bourne. He's a professor in the Department of Pharmacology in that sunny place in the University of San Diego. Many of you probably know who he is already, but he's well known certainly for work on the Protein Data Bank. He's Associate Director, the RCSB Protein Data Bank, or PDB to the rest of us. He's also, of course, Editor-in-Chief of PLOS Bio, and that's a very popular journal now, and is a proponent of free dissemination of science. Hooray. He's published over 300 papers and some very popular textbooks on bioinformatics. So he's also founded 4 companies and received numerous rewards for his work in computational biology and publishing science. Some other interesting facts which I didn't know about. Excuse me. He wrote a book, Unix for VMS, which also happens to be in French. So those of us old enough to remember VMS will know what that book's about. And he also likes to live dangerously by riding motorbikes and flying planes. So I think I'll hand it over to you now, Phil.
Thanks, Herbert, for that embarrassing introduction. Okay, so what I was going to do today is really to tell you a little about this 3D virtual cell project that we've been working on for a while, that's actually funded as part of the S2I2 program from the National Science Foundation. And what I'm going to do is, my agenda is really about that project, but I have to say that the project itself is still in a relatively formative stage, and so I think that there's some lessons that I'd like to sort of bring to the table here based on things we've done before in the way that Herbert described, say, with the Protein Data Bank. I've also been involved in the immune epitope database for about 7 or 8 years, not the 15 years or so of the PDB, but and other community projects. So Herbert also mentioned working with the Public Library of Science and starting a journal with them, again, all around community. So I'm actually going to essentially offer some opinions about sustainability of this kind of enterprise through community engagement, which seems to me to be a critical aspect. So I don't think we can go. And we all have lessons to learn that we know here, and more to learn about sustainability and how important that is to what we do going forward. So I really can't overemphasize that enough, because it's all well to have something and use something, but if it disappears in a year or two, that's to be a big problem. So I'm really going to emphasize that in the context of the 3D virtual cell project, which we're trying to move forward. And what the challenges are there, and then essentially how we address those challenges over the years and other resources I've been involved with. So, it's also a little, I'm a little embarrassed to be talking to this group, because I am not actually a modeler. I've been dabbling to some extent, and of course the definition of that we could spend a lot of time discussing, but for my own personal definition, I would say I'm not a modeler. I've been working with some models, and particularly in collaboration with people like Bernard Paulson here at UCSD. The other, I mentioned communities through PLOS and the PDB. I'm also involved, Herbert's been involved with this to some degree as well, and others perhaps on the call here, or on the webinar. And that is one in particular, Force 11. It's a future of research, communication, and e-scholarship that is really a group of like-minded people, a community, coming together to consider alternative dissemination models and sustainability models around scholarship, of course, of which the 3D virtual cell is a good example. And then, there are other organizations, DELSA and NIF, Neurosciences Information Framework, and the Life Sciences Association that DELSA represents, again around data. So these are all communities that are out there and quite vibrant, and certainly providing input into this. The ones in italics there are the ones I'm perhaps not so involved with personally at this stage, although the neuroscience information framework is really something that forms a good backbone of what we're doing in the 3D virtual cell, and you'll see how that works in a minute. I'm also a university administrator, an associate vice chancellor of innovations for UCSD, and I've only been doing that a little over a year, but I have to say it's given me somewhat of a, I do that half-time, so it's given me somewhat of a different perspective. So I end up fighting with myself a lot because the rest of the time I'm a faculty member. So, but it's actually made me realize the role that institutions have to play in this, in these sort of sustainability models, and which I think is really quite seriously underrepresented at this point. And I'll say a bit more about that in a second. And then just being on various advisory boards around what's going on in this space in terms of modeling, but also more generally community resources, gives me some insights. So, or I should say some biases, which I will have no fear in passing on to you today. So what got me thinking about the 3D virtual cell specifically? Well, the PDB, as Herbert introduced, is a large and well-established structural biology resource that really is a community resource, and I'm going to keep harping back to that. But it's been around for a long time. In fact, we had the 40th year anniversary just a couple of years ago, and of course, most of that predates certainly any pretty extensive. It predates the web, and it predates any, I wouldn't say all digital computing, but a good part of it. But what was interesting at that meeting, Jane Richardson, who's a pioneer in protein structure, representation, and characterization, described the early emergence of hand drawings of proteins, and particularly the iconic ribbon diagram that really is, we all see them all the time. I noticed the new director, John, of NIGMS, in his press release, there's a screen with a molecule, a ribbon diagram directly behind him. So I guess that's a good indication of how sort of prevalent this is. But, you know, interestingly, what's happened is that is now, no one draws those by hand anymore in the way that Jane and Erwin Geist did in the beginning. But you know, that's now all done with some pretty nifty computer graphics programs of various ilks. So all of that sort of moved into the realm of automation. Then David Goodell got up and described some of the beautiful drawings he's done, like including the ones on the top left-hand side there, where he actually depicts either complete cell contents or subcellular compartments. But he's also an artist as well as a scientist, and you know, at this point in time, he effectively does the majority of that by hand. So in a sense, after 40 years, we can draw molecules, but we can't draw cells, and not in a way that's really quantitative and meaningful. And you know, one of the drivers for me as a visual and sort of being rooted in structural biology is to see that kind of thing happen, but on a much larger scale at subcellular or cellular levels. And you know, in fact, there is progress being made in this, and in fact, there are tools within the 3D virtual cell catalog called CellPack, which emerged from David and Olson and others that actually do this. Or I should say do it, but they begin to address the problem. And so, you know, this is the kind of thing, you know, these are, to me at least, the next big steps. And we should be really trying to promote this adoption and development as quickly and as accurately as we can. But when you know, but having said all that, when I think about software, I actually get quite depressed because, you know, what I see a lot of the time is effectively exactly what was happening when I was a graduate student, which is a huge amount of time ago, in terms of how we maintain and manage and develop and use software. So in summary, I looked at this back in 2008 and actually wrote an editorial about it with other folks in my lab. And it's sad to say that many of these resources certainly lack persistence and they lack usability. They cost, they're located in silos if they're available at all. It doesn't really meet the needs of current biology. There's no two ways of questions about that. I mean, resources like GitHub and SourceForge and so forth are definitely aids, but there's still very much to do. And so the idea that we can do better in terms of maintenance and sustainability it just seems to me to be paramount. An example of that, which this article refers to, is we actually tried to reproduce a whole series of methods that actually define domains in proteins. What it is doesn't matter. But going back 20 years, there are methods that have been published in high prestige journals that describe these methods, and which say then the software is available from the authors. And of course, it's that means one of varying things. One, that you get it the next day and it actually works, which is very rare, or after a year of begging and pleading, you still don't get anything from the authors. So to me these are sort of unacceptable situations in the current environment, and there are lots of moves afoot to begin to address these kinds of things. And what we're hearing both from the NSF through this kind of program, and also the Big Data to Knowledge initiative through the NIH, I think encourage me to think that we're on the verge of a pretty significant change here, and I think that is very exciting. So in the end, and some of this is all rather ironic, because in our pockets we have effectively a much better way of doing things, which doesn't really seem to have transcended its way to science in a big way yet. And that, of course, is the app model for computing. Now, you can make lots of arguments about, well, that doesn't fit well or it doesn't apply well. But I think conceptually there's much we can learn from that. Consistent user interfaces, common calling interfaces, and ratings and commentary, and a store of where to get some of this content and some of this software and associated data that goes with it, and ultimately taking that and putting it all together into a modeling environment. So these were sort of drivers that got me thinking about doing something in this space. So that then led to the idea of this 3D virtual cell project, which was really to build community infrastructure to accelerate interdisciplinary science. Obviously, there are some very valuable efforts already going on in this space. The virtual cell project from Leslow and others, of course, in Connecticut is a good example, just to name one. There are a number of others. And so, certainly the intent was not to, but really to try and be synergistic with some of these other ongoing things. And really, which is where the 3D part of it comes in, essentially, in that that's the part that I felt that we could contribute the most to. So that's sort of some of the background thinking in all of this. But when you start thinking about what this should look like in a new paradigm, or there are lots of aspects to this. There's essentially the notion that it becomes an information hub, and I'll say more about hubs in a second. That it really becomes a place for collaboration by a whole series of interdisciplinary scientists, and of course, ultimately, there are at the core of it, there are a series of models and data and software and a whole series of other things. But you can't have really any of all of that without the things on the right-hand side, which are all things that we discussed in the context of this project. So we had actually last December now, I'd say a very motivational workshop around some of this, and I would encourage you, all of the presentations are online and they've all been conceptualized, and I'll show you a couple of those conceptualizations in a minute. But and I think it got us, a lot of us, thinking about these kinds of things. Sustaining that thinking is another matter, but at the time it really takes you forward, and it makes you realize that you can't really do these things without education and training as being an integral part of the whole process. It's sort of learning by doing, effectively, with respect to the contents of whatever it is you're putting together. That rewards and incentives are something that in any field, but in academia we can't ignore. It's really critical to the whole enterprise. And right now, so much of that reward and incentives revolves around publications, and that is rather unfortunate, I think, in the current day and age. And there are, again, fairly early stage developments in various places to address that, and I'll say a bit more about that in a second. And then, of course, you can't have any of this without outreach. Is critical to any of these kinds of things, and I think something that many scientists are not particularly comfortable with, but yet really needs to be front and foremost in thinking as one goes ahead and develops these kinds of things. So, what's the impediment to doing this? Well, I've sort of alluded to it a bit, but let's say a little more. So the notion of hubs, which is really an aggregation point for information, in this case, say around various types of models, but it's more than that. It's really a place to exchange all sorts of information. That, I think, they have worked differently in different communities, and there are examples of things that have worked quite well, even that predate good tools to deal with it. So an example of that, I would say, would be the C. elegans community, and how they gathered together around the sort of common theme. On the other hand, you've got the Public Library of Science that have experimented with hubs, and yet they've had, I would say, mixed success so far. And part of it, I think, has to do with the metrics of what constitutes what's perceived as value, albeit perhaps wrongly. So in other words, you don't actually get rewarded for sticking something in a hub. You get a reward for sticking something in a journal paper. And I think that these kinds of attitudes are very important to change. I think we're going to see a real change in the whole notion of education in the next several years, which actually will help drive these kinds of resources, because education has been very much basically just providing information and expecting people to absorb it. That now is almost is standard fare. Students want much more now. They want the how rather than just the what. And when you have the how, then that brings into play all interaction and, for example, running models and testing models and experimenting with models as part of the educational exercise, which makes all of this all the more valuable. And MOOCs are a first step in taking us there, and we're going to see much more of that in terms of interaction rather than just basic talking head delivery. And so it's all very exciting, really, and that certainly resources like this or others are going to really need to embrace this kind of sea change, in my opinion. Again, just to emphasize the point, the idea that software and data are undoubtedly undervalued. As a result, data scientists and software scientists are undervalued within the current scheme of things, particularly within academia. and improved models of comprehension remain sparse beyond the paper. So the idea is the paper becomes an executable environment to actually conduct the next set of experiments is something that some of us have been pushing for. The Force 11 community wants to see this, and we're making some progress in that direction, but it's somewhat limited still. So institutions clearly have a big role to play in this increasing the value of these folks, and I'll say something a bit more about that. So this is an example of the conceptualization of the 3D virtual cell project. I've said a little about some of this already. Basically, it's any of these kind of projects. This included is really a journey, and ultimately in this case, the end result is to have would ideally be to have accurate prediction of cellular function that's acceptable to all stakeholders. That requires new reward systems, dissemination models, a changed sociology, but the outcomes, obviously, on the bottom right there, could be could be many and significant, and you can almost imagine anything coming from these. So it's a very exciting time. So what all of that's sort of useful philosophizing, but where are we sort of exactly with this project? So we had this conference, so we're kind of in this phase one. We were going to have smaller group meetings. We've actually deferred that because the program itself doesn't have any funding to continue this, at least in this current fiscal year, and that may change going forward. So we've really gone into sort of a mode where we can try and sustain things and grow things with the community. So as a result of that, we've developed the community website and now a resource catalog, which I'll say something a little about. And all of this was based on input from the conference, but also things like community surveys that we've conducted. So in a sense, this just sort of summarizes what I've said before. The idea overall, of course, is to try out some things with the hope that we can do better than we've done before. And then, of course, you can begin to argue how we measure what does doing better mean, and then I could get into some metrics for that, but let's just sort of keep it simple. So, I mean, I think there are different components to all of this. There's particular, there has to be incentives we already talked about. There's how you crowdsource a lot of this. There's the openness for it. There's business models for it, and so on. And I'll say a bit about, and of course, ultimately it has to be fun. And I'll say a little more about some of these things in a second. These points here are things that came out as sort of major discussion points within what was a very open-ended conference I mentioned we had last December. So it was really people themselves defining a lot. There was a lot of open space discussion for people to define what they thought was important to these kinds of initiatives. So, I think the thing that we, and what I believe in personally is, you have to be very careful between, with respect to the biological problem that you're trying to solve. There's this tension between building something and hoping people will come and use it, and then there's essentially only building what is predefined as the biological problem. And we can debate where we sit on this particular line. Some things have been, I would say a relatively small number have been successful with a build it and they will come model. Others, I think the idea of having driving biological outcomes to move it forward are key, and we discussed that in the context of this project, what that means. Is it are we modeling a single cell type? Are we modeling multiple cell types? And what is it we want to solve with these models? What are these this data and the methodologies and when you put it all together, what is the problems we're solving? I'd say, that is still an open question. Another aspect of all of this, which I think I'll come back to because it seems to me to be so critical in all of this, is the notion of trust and what that means. And I'll give you why I think that's been so important in building the protein data bank. So to say a little more about that, I would say that trust in the data over the 15 years we've been running the PDB is perhaps our biggest achievement. And so I think, and I'll show you how I think we got there, but these are to be are absolutely critical components of getting a community buy-in onto a resource. So I would say that just from this experience, I would say that trust is like compound interest. You don't have much to start with, but over time, if you keep adding, it starts to grow more quickly, and ultimately, it starts to compound pretty quickly. But having said that, you can also have a crash, and you have to be very careful that you don't lose your investment. From our point of view, I think, and I'm really speaking from PDB experience here and IEDB experience, is that trust really came from listening to a lot of different people, and essentially all of the stakeholders, both those who provide data and those who use data, and that, in fact, those are also responsible for helping us sustain that data. Comes from engaging the community in every aspect of the process. An example of that is that we form in the PDB task forces whenever there's a particular ticklish issue to be dealt with. The validation of structural information as an example. There are lots of opinions about that, and there's lots of methods for doing that. So the idea is you bring a task force of the world's leaders together to actually provide input and essentially a report, which then effectively gets published, which describes what the resource is going to do in that context. We've done that in a number of different ways, and it's been very successful, I think. Trust is also obviously about the quality of the data. I'll say a bit more about that as well, but and of course that relates also to consistency and level of annotation, and I'll say more about that. And also responsiveness to the user and to that community from whoever is responsible for the resource itself. I think when there's a lack of responsiveness, it really impacts trust. And then, of course, the quality of the delivery service. You've got to be as good as Google, or you're not in business. And so that all goes to build trust. And then I'd say perhaps the most important thing about getting trust is the quality of what's in the archive. Again, just to give you some representation from, so what we think about in the context of the 3D virtual cell really is driven, at least for me, in what's happened in terms of the PDB. So in the PDB, I would say over the last 15 years, we've probably spent upwards of 25% of our budget on remediating data we already have. So, if you're working in a community resource, then you really need to rely on the community to do that rather than budgets. But it's something to constantly think about, and because there's a temporal aspect to this. So what might appear to be a perfect piece of data, of course, there's no such thing, but today is clearly not so perfect tomorrow. And the idea that that data would have new and more consistent, for the time, metadata associated with it is always something to think about. But doing that, you obviously have to have a copy of record, and you have to have versioning and all of this sort of thing. So and then ultimately what's been critical to us, and it's critical, common accepted practice now, but certainly wasn't at the time 15 years or so ago. I mean, remember when we started doing this, no one had actually heard of ontologies in biology. Even the genome ontology wasn't really out there. So we didn't really realize we were building an ontology, but that's what we did, and an associated data model, and that was being critical to everything we've done over the years. And so I think in putting this resource together, that also needs to be front and foremost in many ways. But we made mistakes. I think we made that representation too complex at the time for the community to really embrace. And it's happening now, but I mean, the community has kind of grown into it over the years, I suppose. So certainly learned from that experience is that simplicity as well as accuracy is something to strive for. We can, if there's questions, I can go more into that. So just give you an example of the kinds of things that were talked about at the meeting, which are things that ultimately, some of which have already been added now to the catalog, but some modeling examples. So Marcus Covert, and I think his own work on complete cell modeling from genotype to phenotype, which is in some ways, I think at least in the popular press and perhaps deservedly beyond that, and I'm not the person to judge that, has really helped sort of propel these things forward. And so he talked about his whole cell modeling, and an interesting concept within that was that the interplay between theory and experiment. And as to quote him, never trust a good experiment that doesn't have a good theory. So I think this says a lot for how far we've come, but also how far we have to go. And then just to give you some other ideas of things that were talked about, there's the sensory flow model based on rodent whiskers and the impact, the anatomical visualization of that system and its relationship to the cortex, and how that's being measured in that interplay between again, genotype and phenotype. I think just really exciting stuff. Likewise, Tom Knudsen talked about predictive toxology using virtual embryo models. I mean, I'm sure some of you are much more familiar with these models than I am, so I won't go into any of the detail of it. But if you want, I think, a really good overview of some of these models, if you're not familiar with them or you want a refresher, we didn't let people talk for more than 10 minutes at the meeting. It was a TED-like presentation, and each of these models are presented by these people. And this is just a very small subset of about 30 different presentations in this regard. Andrew McCulloch talked about his cardiac models, which I'm more familiar with, and I just emphasized that part of what he said had to do with training. Clearly, this is something that's first and foremost in the minds of various people that we really need to have more in-depth training as we're putting these sorts of resources together. The resources provide some ability to do that, and we can talk more about at least what that might mean if people are interested. So just to say a little more about communities, Herbert talks about, he could talk more about this afterwards much better than I can, but clearly there are communities out there in this space already that clearly need to be embraced, and there's overlap and so on. But it really has to involve a lot of young people. Just to say a little about communities that have sprung up again to support the worldwide PDB. So I just use this as a sort of representative example. That the slide on the left is just 40 years of people who worked on the PDB who are still very passionate about it, and it's just a subset. And then on the bottom right is the people who currently work on it. But the point is that it is a worldwide initiative, and it's interesting as a model for how it works. So just to say something about that very briefly, is that effectively that there's a degree of collaboration. So there are multiple data input points, in this case, of course, around structural information, currently in Japan, Europe, and the US, although there's new tools being jointly developed by each of those three groups to facilitate additional partners being added to the data deposition. And the agreements have all come, so the joint development of the tools are each funded by the respective institutions. There is no global funding for this. It's essentially the bottom-up, completely bottom-up in terms of having the scientists themselves see the need for working together and recognizing in ways that the funding institutions haven't really at this point, which is that that data is global, and while funding is national, it's somewhat of an impediment. But there's an agreement to work together to move these things forward, and it's actually very exciting. So there's agreed upon sets of standards. There's meetings of the WWPDB to debate new developments in the field, which right now relate a lot to complexity of data and different methods that are becoming available to provide this kind of information and how it should be handled. So there's a scientist-led agreement, community agreement on how this should operate, and I think to me at least, it's a good example, an exemplar for the future. Having said that, when the core information is available, how each of the partners within this organization use that information and the services they offer to their respective communities is, in some senses, a little competitive. And that also, one can argue, is a good thing. So there's a sense of collaboration and competition built in to the enterprise. So I think, again, it's an interesting model to think about in the context of these things. But having said that, in this context, it's not about institutions. I mean, data institutions. That may so there's no real identifiable institution that's really influenced what's happened in the PDB. Now those sorts of things may change in the future, and I would hope they would, because there's a lot of activity around, for example, within CODATA or within the Research Data Alliance, as just two examples, which are fairly broad. And then, of course, there's more domain-based data initiatives going on. So I would hope that that in some ways that they will impact what we do going forward. But it's not had an impact now. And again, another aspect of this which has been absolutely critical is, again, it was the community that created, it wasn't, in this case, it's interesting that now data sharing plans are really, I think, and I'm very pleased to see it personally, are coming from funding institutions. That's not where the data sharing plan for structural biology came from. It actually came from the community who really basically got together and went to the journals and to the journal editors and said, we don't want you to accept any paper within your journal for which the data is not deposited in the protein data bank. And it had a profound impact on the community. And in fact, most of the major journals, in fact, nearly all of them, actually conform to that today. So it's actually increased the public accessibility of this data, the openness of this data, immensely. And so the openness and transparency of all of this has to be there, including in this kind of resource that we're developing now. Otherwise, it's just, in my opinion, just not going to work. And that extends to sort of every aspect of what you're doing, including all of the software you develop to run the repository so what needs to change? Well, I think with respect to the data itself, I think there are sort of some, and this is in some ways beginning to be addressed by different initiatives, particularly the Big Data to Knowledge initiative comes to mind. But I would say that as you start developing this kind of modeling resource, or this, you have to think about the value of the components in it. And I have to say, I don't think we have a good handle on that right now, certainly not for this kind of data, but even for something that's as well established as the Protein Data Bank. We don't really know how the data are used. I mean, that sounds, we know, and we report statistics that we have 300,000 unique users a month, and there are 7 structures downloaded every second. But what we don't know is, why, which structures and why, as in that context. What drives particular data usage, when and how and why. I think we really need to have some more research done on this, and there's certainly some very well used resources out there for which we could get that kind of information. And I think we need to know that because we have to make hard choices about sustainability. The current sustainability model is basically getting more money from the federal agencies to support it. This is not going to scale, and I think the idea that this kind of virtual cell resource, or the resources that come along, how they interface and intersect with the private sector as potential points of sustainability is an untapped and yet potentially exciting avenue. We can go into more on that if people are interested. It also points to reductionism. The reductionism, say, in the human genome is given the redundancy from a reference genome to an individual's genome. Clearly, there are gains to be made there, which are not really being addressed too much at the moment, but I think will come into play. But deciding what the idea of data reductionism, in fact, what to throw away, is something that we haven't really faced yet. Perhaps in a new resource like this, that's not an important aspect, but it will become over time. And then, of course, there's the whole long tail associated with data rather than just the very large data sets. Institutions and all of this, I think, must play a greater role. We need more, institutional repositories, to my way of thinking right now, are effectively silos, and I think institutions could do much more internally and externally to deal with that. I don't have really time to go into that, but I think, in a sense, to develop a community resource that is going to be housed in one or more institutions, those institutions need to, in some sort of ways, play a role for how this happens. And then, of course, you need people to sustain these, and they're ultimately going to have a home. So we need new metrics to actually evaluate these people, and we're actually, some institutions, including our own, are working on this so that we actually measure people's, particularly data scientists, in different ways than just papers. All right, so where are we with the repository? So we have the model repository. We now have a catalog for, and we're kind of measuring community uptake on this. This, I should say, so it's really a repository at this point. And you can go there and you can see, don't see a URL, sorry, but you can easily just Google it and find it. And there's I think about 1,700 items of information in there right now. It also is integrated closely with the Neurosciences Information Framework to pull relevant content from there and also vice versa. So it's a bidirectional interaction. So that's really, we're kind of monitoring that at this time as a sort of phase 2 activity. These are all, I kind of put some of this stuff up before, but these are all potential phase 3 activities, but it's going to need some funding from somewhere to really to sustain these, as well as community, really to help with the community, but also I think basic sort of project management functions that so well now are performed by the Stephanie Hagstrom, who works on this and is just great, as well as the NIF folks under Marianne Montone, for which we're very grateful. But there needs to be at least some core level of funding, and that's going to really depend in part on the outcome of this program within the NSF, and possibly we're looking at other sources of funding as well, of course. So so I just say that, for example, I think the idea of public-private partnerships, which I talked about, clearly have to sort of play a role here. So I think I'll just stop there, because it'd be good to have a bit of dialogue about the kinds of things I've said. I realized a lot of this is more general than about this particular modeling resource itself, but I really think you've got to be thinking about the big picture as you begin to develop a single resource, which the big picture being something that transcends resources. So I'll stop there. I have no idea whether there's anyone still on the line, but I hope there is. And I'll stop. Is there any questions?
Yes, I have one question. The catalog at the moment is really a clearinghouse for all other people's models and software. Is that its purpose? Is it a sort of a central place one can go to find everything that's on the internet?
Yes, that's really its intent. I mean, it's interesting in the context of what's going on with respect to catalogs that are coming from the NIH Big Data to Knowledge initiative. So there was a discussion, and there was a meeting last week, I guess, the week before now, I guess, about a data catalog, and there's going to be a forthcoming meeting about a software catalog. And to me, these are very important developments. And so there will be points of registration for these things. And I mean, what ultimately relies in the central catalog versus these more specialized catalogs is something that's going to evolve over time. But the idea that there are these sorts of at different levels, these kind of clearinghouses, and the ability to go in and comment and rate these kinds of content would seem to me to be just a big step in the right direction. Because, I mean, I'm sure you have this experience, and maybe it varies from different fields, but whenever I embark on some new kind of aspect of what we do, and we try and find out what software, what test data, what models might be available to actually apply to the problem, it wastes a huge amount of time. And anything we can do to reconcile that and make it easier is sort of what the objective is here. But certainly, it was a long-winded, rambling answer to your question, which is, yeah, it's really a point of registration at this point.
I guess its utility will be in how good you can search for things. Because at the moment it says there's 77 software packages, but if I search for them, none come up.
Okay, well that yeah, I have to look at that. But yeah, I mean, this clearly needs work.
Yeah, I know.
Phil, is it possible for you to just show a little bit of the catalog on your screen?
Yep, I think so.
I mean, I wonder if it's better to have a catalog rather than, say, individual models, but to have a catalog of repositories that exist, because the individual repositories will have very superior search capabilities. Right, otherwise you're going to have to build up your own sophisticated search capability into your catalog. So is it better to point to the database or to the repository itself, or?
Well, I think what we're going to see here is sort of a hierarchy of in fact, this already exists, maybe not in the notion of catalogs, but in the notion of, it could be in terms of data content. So, for example, in the case of the PDB, it's really a recognized source of primary data with some tools and around it. But then there's a series of expertise that exist which have done lots of things with it. For example, classification of information. in which case it really becomes the job of that resource to provide the correct pointers to get at that level of information.
But I mean,
but what none of these, what the catalog is sort of one thing, but then, I think you also want to, from that, where it is yet to be determined, but more than likely a remote site, but you obviously want to be able to exercise what it is you find, which you can't really do almost anywhere right now. So in other words, to actually run some simulations, for example.
Well, you can run simulations, but they're just not very good yet.
So
platforms. I mean, there are online services for running simulations. It's just that what you said in your talk was that sustainability is the issue, because these things come and go, and that's one of the big problems in this whole field.
Right. I mean, I think the whole essence here, and I'm not, whether it's going to happen in this case, I have no idea. But I think you've just got to get you have to slowly build engagement, and I think what you do, everything you do, should be driven by that goal.
Yeah.
So right now here's the catalog, and then there's a whole series of fairly cursory based subcharacterizations of what's in it, and you can go to different training things, for example, and then right now these are just sort of resources that register themselves in that way. And then you can go to one of them, and you're just going to pass out. So there's this is very, and in fact the representation itself, what's the catalog is drawn pretty much from what the NIF community provided for us. So it's really about getting enough enough useful stuff there to actually have people tell us, well, it would be much better if. And then have people and then go to the next step. But I have to say that that as I said already, you need to have a core set of people who or at least resource to essentially maintain the base level of this. And given the current state of funding with the SI2 program, it's not clear whether that's where this is going to come from. And I've had some discussion with foundations around this, but it's a matter of pushing forward. So I think again, in all of these, you really need one or more champions to push these things forward. And I have to confess that I've been a little sidetracked lately with some of my other activities. So we have to just see where this goes in months to come.
I mean, we're lucky with the models because the European Bioinformatics Institute has put in a lot of resource to curate and maintain models, so we're lucky there.
Yeah, I mean, these centralized resources, are just, I mean, it's just so valuable to have things like that, but they don't necessarily cover everything. That's the problem.
No.
All right, any other questions for Phil? This has been really useful, Phil. Hopefully we'll be able to help you advertise this catalog through the consortium.
Yeah, I mean, I mentioned outreach is clearly a key part of all of these things. I'm hoping, that the NSF will, get that this whole program will be sustained. I have to say I've had some discussions across agencies with respect to this kind of initiative, and I really believe that we need significantly more crosstalk between the agencies in meeting the end needs of the scientific community with some of these these kinds of things, because there's slightly different emphasis here in the SI2 program than what's going on in the big data to knowledge program, but there's also some real synergies to be had. And it's not clear to me that those conversations are going on.
So thank you everyone for participating. And
yeah, thanks, Phil.
Pleasure. Thank you.
Thank you, Phil.
Bye now.
Bye
Bye.
now.